When we think about the preclinical hub, I think the key message or the key goal is that it is a resource. It is actually a collection of multiple resources that would help speed up drug discovery. Not only speed it up, but also mitigate the risk that comes along with drug discovery, especially in a rare disease such as NF. But it's more than that. It's actually also a network of experts and collaborators that are instrumental in selecting the appropriate tools that could help us speed up drug discovery. With CTF kind of in the middle, orchestrating and making the appropriate connections and providing access to the most translational models, which means that the results you have in the mouse and the cell models represent the patient population. So we want to always be able to take our findings from the models we develop and make sure that they correlate to what we see in the clinic. And those are the resources that we're focused on and will be making and already are making available to the community. And when we say the community, it's really not just pharma partners, but also the research community and other institutions. So this is really for the NF community to use as they need in order to accelerate drug development and to find those next therapeutic agents for NF. That is the goal.
Do the clicker. Hey everyone, I think I'm on mic, yeah. So I don't have a clicker yet, so I'll take this time just to introduce myself. My name is Miranda McManus. I am the parent of a 21-year-old, almost 3 days with NF1. And I also am a molecular geneticist, so I kind of have a leg in both the parent community as well as the research community. And one thing that I've really noticed recently is that with social media and everything that is out there, we get so much information so early in the process that most people never saw before. And so I wanted to try to use today to give you guys the tools to understand the research process a little bit better and hopefully figure out how you can be involved if you want to, all right? So you see posts all the time. They're all over the place. Lots of times they're very reminiscent of a miracle cure, right? And it's great that we're getting this information so early, that science is shared so quickly, especially with the NF deserts that we have in this country and around the world. It helps people to empower themselves and advocate for themselves. But the problem is that we're getting a lot of it very, very early now. Things that may never show up in the clinic, things that may never work for NF, tend to be on our Facebook page the day after the paper comes out, right? And that can, be a problem because early access gives you that information, but it can also lead to some false hope, right? And so understanding how to work through this process and understanding that in some cases, things might not that's the wrong button. Things might not ultimately ever apply to NF or schwannomatosis, or that they might never make it to human trials. It doesn't mean you should give up on hope, but it means you should understand a little bit more what step in the process that information is at. So that's my goal today, is to try to give you some of those tools to try to help you learn how to interpret that information so that you have a better sense of what is going on in the scientific world around us, in the world of NF, Schwann, and all the other disorders that are out there. So I'm going to start off with an example that most of you have probably seen if you're in any kind of medical Facebook group or random space online. And that was an incredible story of the world's first bespoke, which just means custom gene therapy, that came out in early 2005. There was a baby at CHOP, the University of Pennsylvania's Children's Hospital of Philadelphia, which you've seen some people here from there. And this child had a urea cycle disorder. It's a metabolic disorder that causes fatal levels of ammonia buildup. And what was really amazing about this case is that within 6 months, they developed a custom gene therapy, a CRISPR therapy, that corrected this mutation, this genetic variant that was causing this problem, right? And fast forward a little bit, the treatment worked. Baby KJ is doing great, metabolizing proteins, hitting all the major developmental milestones, and is at home after many, many months in the hospital. So it made it all the way to the clinic for baby KJ. But this was a very, very specialized treatment. While I saw it all over the place as a miracle cure, genetic diseases, that's going to be an end, that's not exactly the reality of things. Now, tools like CRISPR, fantastic, right? We've got tools now that we never had before, and they're promising. There's tons of promise there. But with baby KJ, this is never really going to transfer to NF or Schwann. Now, it doesn't mean that the things that they learned can't be used later on and carried over, but this particular case was a very specific genetic variant, and it was targeting the liver. And they used lipid nanoparticles to deliver this. Lipid nanoparticles, you may have heard before, they were in the messenger RNA COVID vaccines. It's a delivery method to get, in this case, DNA into cells, right? But the thing is, lipid nanoparticles are naturally processed by the liver. So if you're trying to target the liver, lipid nanoparticles are a great delivery tool for that therapy. Whereas in NF, we've got a nervous system disorder, right? NF, schwannomatosis, both associated with the nervous system disorder. And that means that to have a systemic treatment, you're going to have to cross the blood-brain barrier, which is a challenge and something that lipid nanoparticles are not able to easily do, right? Also, like I said, this is a very specialized treatment. N of 1 specificity is the term that you'll hear. There's N of 1 trials where there's literally only one person in it. This therapy is specially made for baby KJ. Now, does that mean that they could use this platform and make it work for some other mutation? Sure, but it's going to work within a very small realm, right? So while this was touted in many cases and a lot of people got hopes up for like a cure tomorrow, it's not going to directly translate that quickly, right? So I want to talk a little bit about how treatments make it to market. And we started off this summit with that. If you were at the talk the first day with Annette and Frida and Michelle, they were talking about the drug development process a bit. So we're going to go through that again today. We start off with that target discovery. So target discovery is just when you're trying to find the compounds and the specific target that you're treating, right? So depending on what they're trying to treat, there's going to be a specific target that is being impacted, and there's going to be a specific treatment that is impacting that, right? This can take a while. There's all kinds of different processes to come across this. We're not going to really get into it today. But realize that this is the early stage. We don't often hear as much about stuff in this stage. Preclinical development, we're starting to hear a little bit more about. So I'm going to focus more heavily here today. This is before treatments get into humans, right? Before we put any kind of treatment into a human, you usually want to understand a few things. Is it safe? Does it work, right? And then How much does it work? Does it work better than what we already have? And so preclinical development is us trying to get to that point that we're comfortable to start into clinical development and those clinical trials, right? And then after clinical trials, if a treatment makes it through, then it goes through regulatory review. So in this country, we've got the FDA, Food and Drug Administration. If you're in Europe, it's the EMA. Around the world, there's different organizations, governmental organizations that oversee this regulatory process. And then even after approval, you guys have heard at the summit about trials that are going on with selumetinib, with mirdametinib. They're approved therapies, but we're still trying to learn more, right? So there's post-market monitoring, both looking for long-term side effects, but also just trying to understand generally how something works over time and doing comparison type of trials. Does it work as well as what we already were using? So I'm going to focus in on these two today, right? And I'm going to start with the part that you're probably already a little bit more familiar with, the clinical development, so the clinical trial process. So you can break down this process into multiple phases. For clinical trials, which is when we are testing a treatment in humans, we usually have 4 phases. There's phase 1, phase 2, phase 3, and phase 4. Phase 4 is usually post-approval. All these others are leading up to that. And this top bar is showing you the time, who participates, how likely it is to move on from that stage, what we ultimately end up learning. When we're looking at phase 1 trials, that's generally just looking for if it's safe. We look for the maximum tolerated dose. We look for things that make sure that people are able to take this treatment safely. We're not so concerned yet about whether it's working or not, because it's just assessing the safety in humans, because this is usually the first time in humans. For phase 2, we're focused on does it work? And you'll see on trials, there's 1A, 1B, 2A, 2B. These can be split into slightly different stages, right? And sometimes they're lumped together. Sometimes you'll see a phase 1-2 trial or a phase 2-3 trial, right? And that's because this is all a process. You're moving to the next one when you're ready to, right? And so does it work? By phase 3, we're expanding it to a lot more people, where in phase 1, we start with either healthy volunteers or a very small population of patients. In phase 2, we expand that a little bit, still pretty small. Phase 3, we get a lot larger, right? Does it work enough? Is this working across the population, or is it in this small group of people only? And then once you get regulatory approval, if a treatment makes it through all these stages, then there's all this post-market monitoring, and so there could be phase 4 trials associated with a treatment after approval. So just to start off with a couple of case studies, a typical timeline is a drug that you've seen advertised around a little bit here, Koselugo. This was our first approved therapy for NF1. It was initially approved in children. Now it's for children and adults. And this was a long road to ultimately develop this treatment. In the early 2000s, the MEK inhibitor compounds were discovered. So what Koselugo does is target a particular part of the NF1 pathway and shuts it down, at least partially, right? And they discovered through early work that these compounds might be something that works for NF. It wasn't until 2011 that this was actually in clinical trials. So there was a long lead-up period before the first human ever saw a dose for NF1. Those lasted through to 2019. In April 2020, Huge news, this is our first approved therapy for NF1, treating plexiform neurofibromas in children. And finally, that hope that had been building for so long for these compounds became a medicine that people could access. At the time, only children, now adults, now in many places around the world, not just here, right? But it was baby steps to get there. And since then, we've had ongoing clinical trials. You've probably heard about several of them at this summit that are trying to understand better how this works, how well this works, what situations it works in, etc. So ultimately, we're looking at 15 plus years of development for a typical beginning to end type of situation. And realize that every step along the way, you have to have the interested people getting involved. You have to have funders. You ultimately have to have a drug company that is willing. There's a lot that goes into each of these processes other than just the trial. So what you've also heard a good bit about at the summit is that sometimes we can shorten these timelines. And our other approved medication for plexiform neurofibromas and NF1 is mirdametinib, the brand name Gomekli. And with this was a shelved Pfizer asset. This was something that had been put down in the basement to, at the time, the money wasn't worth it to them to spend to advance this drug. It wasn't able to do the things it wanted to do at the time. This happens to a lot of medications. They end up in a basement somewhere because they don't make it through those first stages. But this is now approved for NF1. And when it was able to actually go into clinical trials, it was able to skip the beginning and go into phase 2 because the safety trials had already been done on this. So there was minimal safety work because that had already been accomplished. And so while you still had a lot of the previous timeline in there, phase 2 was where you got to start at the clinical point, which cuts out a significant amount of time by not having to do those initial safety trials. Also got the drug in more people to start out with, right? We also have brigatinib. Brigatinib is another example of a drug with a shortened timeline. So brigatinib is for NF2 schwannomatosis. This is a repurposed drug that was already approved for lung cancer. It is now not approved in NF2 schwannomatosis, but it is in the clinical practice guidelines for NF2 schwannomatosis, and it can be given off-label because it is an approved therapy for another condition, right? Brigatinib was also able to start in phase 2, so it cut those timelines off. So you can see why CTF is very interested in trying to shave some time off along the way and able to get further into clinical trials quicker. There's another drug that's in clinical trials right now. It's just has HLX1502 for a name that we know at this moment. But this is a drug that is previously approved in another country for urinary tract infections. Again, there's safety information on this drug. It was able to move into clinical trials and ultimately move into phase 2. The interesting thing about this drug is that where the time was cut out was more so in that target discovery range because this was identified by AI as something that might work in NF. And early preclinical studies showed that it was pretty effective for plexiform neurofibromas, and so it was able to move straight into phase 2 clinical trials. So shortened timelines can be a big deal. To put all of this in perspective for time frame and the amount of compounds that we have, that drug discovery stage, there's about 10,000 compounds that might be identified during this part of the process. In the preclinical stage, which is when the research is being done in some organism other than humans or something human adjacent, and that can be like 3 to 5 years. These are all ranges. They can push in either direction. And then the clinical development stage takes about 5 to 7 years. Regulatory approval takes about 1 to 2 years. And over this time, that 10,000 compounds has littered down to just 1, right? So anything in those early stages, we have to take with a hopeful grain of salt, right? Because there's a lot that doesn't make it all the way through to clinical development. Only 1/5 are gonna make through that stage, right? But from preclinical to clinical, you see that that's a huge jump. So we're gonna talk about the reason for that a little bit and focus in on that early process of preclinical development. Okay. And in the preclinical stages, since we're not using humans, we need some sort of preclinical model that we can use for early testing. And so that's what I want to make you guys a little bit more familiar with here today. And what do I mean by model, right? So this is in Yosemite National Park. And if we want to somehow model this, we need to have some representation of the object that is meaningful to us for what we're trying to do, right? So the Wikipedia definition for a model is an informative representation of an object, person, or system. Well, nothing is going to perfectly mimic humans, right? So we want it to map back in some meaningful way. So using Google Maps that we're all familiar with is an analogy here. And some of these slides have been borrowed from people that I have seen them from myself that I thought would be useful for this population. And this is from Dr. Carleen Riley at the NIH. This map maps back pretty well. You can see Half Dome represented there. You know where it is. So if that's what you want to know, it's a pretty good model if you just want to know where it is in respect to everything else. But you don't have a lot of information, right? It may not be enough for you. I spent the last week hiking in Wyoming, and I probably would want to know some terrain information on that before I went walking up to Half Dome thinking it was flat, right? And so we can step that up another notch. And we don't need all the features of the original. We just need the ones that are going to be most useful to us, the ones that we're trying to pay attention to. And it needs to be some kind of pragmatic stand-in for the original. But what you need to realize is in preclinical models, the more features you add, it comes at a cost, right? And some features are not that easy to ever really add, right? But I've got the file sizes for each of these just to give you an idea of the cost in a way that you might be able to imagine, right? As you get that satellite view that gives you a little bit more information, is taking a lot more of your storage up, right? It might not be something you're able to pull up when you're on the trail with low internet or something like that. So why do we use models in preclinical research? Well, we don't want to put it into people yet, right? So we want to predict that toxicity before we put it into people. We do it to try to understand the underlying biology. And ultimately, we want to know whether it works, right? So we'd like to have an idea of all these things before we put it in the first person, because why risk the safety if we're not even sure if it's worth it, right? So I'm going to talk a little about some of the common model species that we use. Some of the next few slides get a little bit more in depth than others. I tried to balance it out between the really high view and giving some more details to those of you that may want them. So don't check out if I lose you for a second. You might be able to get back in right on the next slide. So the one that you've probably heard a lot about are mice. Mice are pretty common lab models. They're a workhorse. They're short-lived, which in some ways can be good, in some ways it can be bad. You know, a short life doesn't really mimic the human lifespan. But when you're trying to do things quickly, you don't want to have to wait a human lifespan either, right? But they have different skin, bone, brain biology than we do. Vestibular schwannomas, which are incredibly important for NF2, don't form very reliably in a mouse model. And so they're not perfect, right? We use them a lot, but they're far from perfect. Zebrafish are another genetic model organism. You might be able to see some more similarities between you and a mouse than between you and a zebrafish, right? At least with mice, we have many of the same body parts. That's not necessarily the same in fish. But they're fast and really good for screening. They're very distant from humans. In NF1, there's a lot of duplicated NF1 genes, which is, not ideal because it's not the same. And again, their anatomy just doesn't match us that well and doesn't match the tumor types. Fruit flies. Fruit flies are great for learning about gene pathways. They can be pretty good about learning and behavior as well. But many of the tumors in NF and schwannomatosis are going to have Schwann cells involved, and fruit flies don't even have Schwann cells. They don't have true nerve sheath tumors. So you're very limited in what you're able to do within these models. For some things, they're fantastic. For other things, not so great. The pigs or mini pigs are huge for our community because they do mimic some of those things a little bit better. Pigs are closer to humans, but they're costly, they're slow, they're in very few labs. People tend to care a little bit more about pigs than about mice, with animal testing concerns that, are valid. That's a little bit more of a challenge to sell the pig. You don't want to put everything through the mini pig, right? And people don't have enough money to do that. And even if you do, they're still not exactly the same. They're still a pig and not a human. And then there's even work that's done on yeast. So yeast are just single cellular little fungi. We know from bread and beer, but they're used very commonly as a genetic model organism. But that's just looking at basic biochemistry. They don't have any higher level structure because they're just a single cell, right? So to talk a little bit more specifically about some of the challenges that we see that are specific to NF and Schwann, models tend to be very tumor focused. You probably heard me say tumors a lot in the past few slides, right? And that's because a lot of our model organisms are based on tumors. But that may not be addressing the things that we care about the most, right? Tumors are obviously something that are important to this community, but there's a lot of other important things. And the size of the tumor, even in the case of a tumor, doesn't necessarily translate directly to the functional effect, right? So things like cognitive learning disabilities, bone problems, pain, appearance differences, these things are a little bit harder to model in a lab-based organism, right? Also, these conditions are highly variable. The genetic terminology that we use is variable expressivity. What that means is you can have the same genetic variant, even among a family, and have very, very different presentation. The things that you show are completely different from what somebody else is showing. This person has tumors, same mutation. This person has no tumors, but may have, severe cognitive challenges. Also, there's a lot of other genes involved, rather than just the genes that are associated with giving you the condition of neurofibromatosis or schwannomatosis. There's lots of modifier genes that play a role as well, right? And so that variability is challenging and can be difficult to mimic in a model. Another thing, NF1 has a massive gene size, especially when we're thinking about things like gene therapy. It is a very difficult gene to work with because it's hard to even dissolve it because it's so large. When you're working with something in a lab, even just solubility of it is pretty important, right? So that can be a challenge. these multi-system symptoms, these mutations don't just affect one thing. They affect lots of different symptoms. And even though we have progressed a long way in the 21 years of my son's life, we are still far, far away from understanding the why behind all of these symptoms. And also symptoms develop over time. So like I already said, with mice particularly, they're short-lived. You don't get the whole lifespan. So while condition might develop over decades and different manifestations develop over decades, the animals in the lab actually only live for months. And another thing that we see is like with NF1, plexiform neurofibromas have the chance of malignant progression into something that we call an MPNST. And there's a middle stage. And very often, that whole process is not replicated in the model organism. And so if you're trying to figure out how to stop the process, and the process is different, how well is that going to work? You never really know until it goes into humans. The NF and schwannomatosis genetics just work in general against simple model organisms. One thing that we see in both NF1 and all forms of schwannomatosis that we've identified a genetic cause is that there is a random second hit mutation. What that means, all of us that are born with NF1 or schwannomatosis have 2 copies of each of our chromosomes. One comes from mom, one comes from dad, right? And one of them is mutated. It has a genetic variant in the case of having the condition. The other one's going to be normal because it turns out that the proteins that are made that come from those genes are pretty important. And so if both of them are mutated, and so you're not making any of an NF1 neurofibromin and NF2 Merlin and the different proteins that come from those genes, then that's going to be problematic. And that's when we see tumors and things like that develop, right? But if that's there from birth, you're never even going to develop, right? The pregnancy will never come to fruition. And everybody that is born with NF or schwannomatosis just has that one copy in all their cells. But if you develop something like a tumor, there is we call it somatic. It just means during the lifetime, second hit. So say one chromosome was mutated, now both of them are, right? And that can cause a lot of different conditions. So this is NF1 on the left, schwannomatosis on the right. Probably a little bit hard for me to see from there. But everything that has 2 asterisks beside it are manifestations that are caused by a second hit. The ones that have just a single, so like cognitive impairment, is something that comes along with just that original hit in some cases. But we don't see tumors that don't have that second hit. That's really, really hard to mimic in a model. We also have mosaic and segmental forms. So what happens with mosaicism or with segmental is that mutation happens in early development before you're a fully grown human, like even in the womb. It happens sometime very, very early on. And so it impacts some cells, but not others. So some people with mosaic NF or SWAN, they might ultimately go get genetic testing and have it come back as negative, even though they're showing clear signs, because they might have to have specific cells tested to be able to figure that out. And the test they took was from the ones that didn't have the mutation that was present. There's also thousands of different variants. For the people in this room, there may not be any that have the same variant. For the people at the whole summit, there may not be. It's a huge gene. There's lots of different places in NF1 for it to happen. The schwannomatosis genes aren't large, aren't small either. There's plenty of places for mistakes to be made. And so if we're targeting with some type of gene therapy, for example, what are you targeting? There's so many different variants that are out there, and one treatment isn't going to work for all of them, right? In NF and schwannomatosis, we also have biology that lives outside of the tumor cell. So just looking at those tumor cells is not enough. There's a tumor microenvironment that we're understanding more and more how very important it is. And this tumor microenvironment, there's immune cells, there's mast cells, there's fibroblasts, there's blood vessels that all live outside of the tumor cell. That in the human, that's going to exist. In the models, it's not going to be replicated in the same way. So, a lot of stuff on this slide, a lot of random letters. These are a bunch of the different types of preclinical models that we see in NF1. I've got little icons up there, so this is another borrow from Dr. Carleen Riley. I love these little icons that she had. The fruit fly is the orange, there's a yeast that's yellow, you see the pigs in gray, the mice in blue, there's a zebrafish over there in green. One thing I want you to notice is that most of these are for tumors, right? We've got learning disabilities over here that's a little different. Skeletal dysplasia is bony, but there's a lot of models that are very focused on tumors because those are the easier things to model, right? It's challenging to model some of these other features that matter to families. And I stuck a box around learning disabilities over there because we do have some models that we're starting to look at that now. But it can be a little challenging to figure out how to measure that. Some limitations specifically of NF1 models. Most of the models don't produce cafe au lait spots. Not something we care as much, but you're missing something if you're not developing cafe au lait spots, right? There's something that's supposed to be there, and they aren't there. Skin neurofibromas, cutaneous neurofibromas, they're pretty difficult to model, right? Mice very rarely grow them. And their appearance and that growth over the lifetime, when we can get them to grow, they're either there or they're not. You don't get that random appearance, right? So understanding the process isn't as easy. Plexiform neurofibromas are probably our best modeled tumor, but mice need that engineered second hit. And the supporting tissues, that microenvironment, is only partly reproduced, right? They're working harder and harder at working on that aspect of it. The development of a tumor. Models impose when and where a tumor forms, but that random arising, that just showing up that happens, doesn't get modeled in the organisms, right? So it's the whole development process that we can't capture. We can capture the tumor, but not getting to the tumor. Oops. Some others, like I said, that in-between step from plexiform neurofibromas to MPNSTs, difficult to capture, right? So models will jump straight from plexiform to MPNST without that in-between step that we call an atypical neurofibroma or an or whatever, you'll hear that said lots of different ways. In malignant tumors in MPNST, the genetic changes are more complex. There are other mutations that are happening, other genetic variants occurring outside of the NF1 gene that have something to do with this process. In optic pathway glioma, the models don't model vision the same way that it happens in children. So there are, mice models, and they're doing, and if you were in the session yesterday with Dr. DeBlanc, you may have heard me ask a question about this. They are actually looking at vision in mice now, but the way that an OPG affects vision in mice is different than in humans. In humans, sometimes a tumor can grow, the vision doesn't get worse. The tumor can shrink, the vision doesn't get better. It's not necessarily a direct relationship. Whereas in mice, it's pretty direct, right? If your tumor's growing, the vision is getting worse. And so again, it captures a part of the picture, but it doesn't capture some of those outliers. It doesn't capture the whole picture. Bone, super challenging. We don't really have very good mice models for dysplasia. There's lots of people working a lot on trying to get better models for this right now, but human-type pseudoarthrosis and scoliosis are not as well produced. And then learning and attention differences, we do have some models, but again, how do we know if a mouse isn't learning, not paying attention, those measurements, something can get lost in that. The way that it is typically done in mice is this thing called the Morris water test, and they give the mouse a visual cue, and there's this little platform, and this will be in like dirty looking water or milk, and mice don't like to get wet. And so what they'll do is time how long it takes for the mouse to get to that escape platform, because it's getting there as fast as mousely possible, right? And so if there are cognitive difficulties, you might expect the route, if you're improving that, you might expect the route to get a little bit straighter. Another thing they do with these Morris water tests is teach it to mice over time, so you can think you can't do this too many times in a row because the mouse figures it out, right? So there's a lot of challenges that go along with it, but it is an effort being made to try to address this very important issue to families. So looking at some preclinical models for Schwann, this is covering NF2 and the other schwannomatoses, and you can see that we are sorely behind in that regard. There are not nearly as many models. We've got some models for molecular function. So if you're lucky enough to have your specific type of mutation, whether you have an LZTR1 or a SMARCB1 or an NF2 associated form, then there are some models for molecular function, but notice those are yeast and fruit flies. And then we've got some models for schwannomas, meningiomas, getting there a little bit with vestibular schwannomas and hearing loss. How do we test how a mouse hears? You know, again, you can see when we're not looking at endpoints that are just tumor size or something, it becomes a lot more challenging to measure. So some of the specific limitations that we see in schwannomatosis models, the peripheral and spinal schwannomas that people can get have very few faithful models. In the forms that we're aware of, SMARCB1 and LZTR forms, we basically have no models that reproduce human tumors. Like we can look at the molecular function, but that crossover isn't there. Vestibular schwannomas are probably the largest concern to most of the NF2 related schwannomatosis because of the related hearing loss that can occur, facial paralysis, all of the things associated with it. Mice don't very reliably grow tumors there, and hearing loss and things like that balance are also very challenging to measure. And then in other forms of schwannomatosis, the non-NF2 related forms, tumors tend to scatter along the whole body, right? Nerves everywhere. They spare the hearing nerves, and that tends to be very difficult to reproduce in models as well. So there's a lot of challenges in the schwannomatosis space. Some of the other things that people may care a lot about in schwannomatosis, pain. That's a primary symptom for most patients that have non-NF2 schwannomatosis. We also see this in NF1, right? Pain is something that is pretty important across this community, and we don't have a very good way of measuring this in preclinical models. Meningiomas and ependymomas, which are typical of NF2-related schwannomatosis, again, more tumors that are inconsistently reproduced in the models. And again, we have the same situation with complex genetics, right? There's a lot going on, and that the multi-step processes tend to get oversimplified in all of the models. One way that we measure pain in mice is by measuring paw licking. Because if you deliver a painful stimulus, mouse tends to start licking their paw. They lick it for longer if it hurts more. And so when I talk about oversimplification, I'm not joking. We are quantifying pain by how long a mouse licks their paw. I mean, it's great that we have some way to measure it, but you understand some of the challenges that we have in the preclinical space, because even something that can work kind of well as a measure, it's not capturing the whole experience, right? You're measuring a behavior associated with a specific pain, but it's not capturing everything about that pain, not the way that we would capture it in humans. So I also want to spend a little bit of time talking about some of the new approaches that are out there. So new approach methodologies, as defined by the FDA, are innovative testing methods and strategies that are used to assess the safety, efficacy, quality of drugs, biologics, and other FDA-regulated products while reducing or replacing animal use. So it's been a big push from the FDA to try to move away from animal models. There is, some places where we can move away for sure, but you realize that what we lose just going to a different animal, you're going to lose even more if we don't have a living organism that we're working on, right? And so while these can be part of the process, the animal models aren't going to go away anytime soon, because we have to have something that's a little bit closer to humans. But just to talk a few about some of the different types of NAMs, they're abbreviated. So we've got 2D cell cultures, and we've been culturing cells forever, but now we're able to do it with patient stem cells. We can use what are called induced pluripotent stem cells that I'll talk a bit about in a minute. We can turn them into Schwann cells or nerve cells, and we can use it to watch how drugs behave and to test drugs quickly. So induced pluripotent stem cells are cells that they can take, like even from your skin, and they can, in a lab, introduce some reprogramming genes and convert them back to an embryonic form, then introduce another little recipe and let them turn into the types of cells that you need, right? Huge developments in this space. There's lots of different types of cells that we're able to turn these cells into. There are challenges with using these as well, but the nice thing about this is it can be matched to a specific patient. It's with your genetic information in it. And it's basically setting the time back on your cells, setting the clock back to the beginning, and then redeveloping them into something else, right? Huge technology. Somebody won the Nobel Prize for this. Took a lot of work, but there's still a lot of challenges with them. They're being used in treatments, for example. They can cause cancer, right? So this is right for now primarily a laboratory technology. There's also some 3D forms. So there's 3D organoids and spheroids, as well as what we call organs on a chip. And basically we can grow little like mini tumors. We can take cells, grow them into more cells, and now we've got a 3D form rather than a 2D form. With organs on a chip, we have these little devices that have channels running through them, like this, where you can add some supportive side. You can have some fluid flow, so you can try to emulate a specific symptom. And then you can see that some of the examples that have been used, the liver chip was used to study liver-like metabolism of drugs, and it correctly identified 87% of drugs that cause liver injuries. So this is not the answer for everything, but we care a lot about the impact of drugs on the liver. And so to be able to understand the impact that it might have on liver and be able to use human cells to do it can be beneficial. 3D bioprinting, we can also 3D print cells and build tissue-like structures. Cells organize into tissues, which organize into organs. And so again, it's just trying to create some of that upper level structure to get a little bit closer to human. And there's also what we call in silico models, which are computer and AI models. And these are trained on biological data, and they're able to predict ultimately how genes, proteins, or drugs will behave. We've also got a couple other things that I'm just going to go through quickly. We could take human tissue out, put it in a dish, test drugs on that tissue. We call that ex vivo, outside of life is what that means. High throughput cell-based screening. We can take lots of cells, and we can do robotic screening, and high throughput just means you're putting a lot through, right? So you're doing a lot of the time. We could do microdosing and imaging in human volunteers, so just small doses. And then if you're trying to study things like how the drug moves through the body, which is pharmacokinetics and distribution, where it ends up going, you can label it and do PET imaging to identify where it's traveled in a certain amount of time. So these can be useful. And then there's refined in vivo methods, like using humanized animal models. So to talk a little bit more of that, we can actually take those iPSCs, those human cells. We can create what we call patient-derived xenograft models, or PDX models, where you can actually take human tumors and grow them inside a mouse. We can also replicate the human microenvironment a little bit better doing this. And so this is combining basically one of these NAMs with one of the things that we've been doing for a while, working with mouse models to try to make a humanized mouse model. So now you've got a model that, you know, we're pretty good at working with, but it's a little bit more like a human than it was before. But there's limits. Cells are just cells. The iPSCs tend to stay young. They can vary a lot. They lack real tissue structure. With the organoids and spheroids, these 3D methods, they can recreate some of the tumor biology, but there's no blood supplies, there's no nerves, no immune system. Same with the organs on a chip. You know, we can add some flow, we can add some microenvironment, but we're still, you know, very limited in what it's able to accomplish. Computer and AI models, they can only work off of what they're built on. Can't predict biology that we don't understand, right? It's all based off of what you put in it, so you're not going to get out more than we already know. It's just able to process that information faster. And then with NF1 and Schwann, there's, you know, all these limits of these more meaningful endpoints that are not necessarily measured the best in the models. So what do you guys do with this, right? When you come across something, when you come across a story, when you come across this hopeful piece of information, don't lose hope, but think about the reality of the situation, right? Is it in people yet? How long is it going to take? Which part of the problem? Most of these diagrams that I showed up here, you'll be able to see this talk online, but you can also find them with a quick Google that will give you an idea of how much of a time frame if it's at a certain point in the process. Can you contribute? Do you want to contribute? If there's, you know, something that you want to get involved in this space, ask. Ask people that are involved in the research, is there a way that I can help this? Is there a way that I can help advance the science? And ultimately, when you're trying to process information that you don't really understand, talk to your specialist, reach out to people in this community, find answers from people that understand a little bit more about it. Don't go off on your own and try to understand too much when, you know, everybody that's here hopefully is developing a little bit of a support system, and hopefully that you can find yourself a provider that can help with that as well, right? If you don't already have one. So the last thing I wanted to talk about briefly before I take a few questions is the preclinical hub. If you were watching the video before this, Dr. Irene Morgenstern is the director of preclinical development at CTF, and they have developed a preclinical hub that you've heard about a few times in different spaces here. I'm not going to get into all the details of that. I'll let you talk to Irene about it if you want to learn more. But one thing that they're doing is trying to bring, everybody together in this discussion, patients, clinicians, researchers. So you can scan this QR code to learn about more, but also they're specifically trying to get samples from patients to develop these types of PDX models and iPSCs. And so CTF is actually recruiting individuals from across the community to help generate these new patient-derived models, and ultimately try to better understand. Participating, you're just gonna have to do it at home. CTF provides a collection kit. You can get the blood drawn from a local phlebotomist. CTF will cover any costs that you end up encountering through the process. And if it sounds like something you would be interested in, reach out to Irene. This is her email up here. She'd be more than willing to talk to you about it. And with that, I'll take any questions that you may have.
It's a little morbid, but
yeah, that's all right.
Is there a way that we, with NF, can donate our bodies for research?
So yes, there is. I do not know the details of that, and I don't know in the NF space specifically who's working with that, but I know that there are many ways to donate your body. I can figure that out and get back to you if you're going to be around later today. I'm sure there's still somebody here that is. Does anybody in the room know? Yeah, but there's probably a way. It may not go directly to NF research at this stage, but it's definitely something that should be thought about. So, sorry, I don't have a better answer.
I think, is this on? I think for donation of your body for research after you pass away, I think you can do that through estate planning.
Yeah.
And specifically mention that you want your body to be used for research at, a local medical school or something like that. I think that would probably be the mechanism.
Yeah, most likely.
Yeah.
Most likely, but I don't know what would directly get it into an NF. I don't know about for the NF research community how that works.
Hi. What can you tell us about early stages of research around fixing the problem at the genetic level? All summit, I've heard tons of information about fixing the symptoms of the problem, but I haven't really heard anything about fixing the underlying genetic cause.
So I will say that I was at the research conference last week, and I was thrilled with the amount that I heard about prevention for the first time ever. Patients, it is incredibly important too. And it's hard to think about how to work with prevention when you aren't even sure if somebody's going to develop something, right? And so there's a lot of work towards starting to think that way, because you have to measure different things when you're thinking about prevention. As far as starting from the beginning, eradicating things, gene therapy of some sort would really be the only solution, right? And at this point in time, nobody's working on any germline gene therapy, so it wouldn't change your original genetic mutation, right? That genetic variant that you had from the beginning. But it could potentially change it in the cells that matter going forward, right? So it might not fix the problems of the past, but you might be able to fix some of the problems of the future. There is a lot of work going on. The RAINS collaboration that is focused on clinical trial endpoints has a gene therapy working group. I actually personally just completed a patient preference study for gene therapy. Hopefully some of you guys in this room filled out that survey that came through the registry, but it made us learn a little bit more about what patients want, and we're communicating that back to the researchers and the scientists that are doing the work. All the people that are working on these animal models are very engaged in the process. So it's a bit down the road I can't say when it's going to be, because when the break comes, it's going to come, and then who knows how quickly things happen after that. But the work is being done. It's just a lot more challenging, and with all the different, genetic variants that all of us have, that's challenging too, right? Because the solution for one isn't necessarily going to be the solution for all. But it is being thought about, I promise you there. It's just a much longer road. And really, all the genetics are just getting sorted out.
Hi.
Hey.
Piggybacking off of what she was asking, so there's an NF registry. Not everybody in the registry has had a DNA test, but presumably there's some cross-referencing, right? So my son had a DNA test, and I know that his mutation, they gave me a number. It's on like 1,480 spot, of the NF gene. If you guys were to look at this ginormous database and say, oh, we've identified 5 people who have that mutation and nothing else, just that one. Do they have, he's going to be 20, do they have comparable medical histories? If a new family came and their kid had that exact same variant, could we maybe say, without scaring them, but these are the potential medical scenarios, because now we have a population that we've measured that we can group a little bit?
Yeah, so that is something we've actually been talking about a good bit in the range gene therapy group, because we recognize that there's going to be a specific group of people that are going to be eligible for the therapies, and we don't know which ones those are yet. And we're still trying to kind of work through that problem, but there's a lot of people that have genetic tests separately. And so we've even thought that if people were able to upload their own genetic tests so that then they could be processed if they were willing to do it, then that may be a way to, is Kate in the room, Celts? I think that they were adding to this version of the registry, trying to add the capability to add the genetic information. I'm not sure if that actually happened or not. But do you know, Jamie? No, okay. But it is being talked about. It's not sure exactly how to work that out, because you're dealing with personal health information, traveling across, and so it's a little bit more challenging of a problem. But I think a lot of patients would probably be willing to upload if it was something they might be interested in, so that might be the better solution in the long run. Anyone else? I think we're running out of time. All right. Well, thanks everyone.